In this video, we are going to be describing mitosis. We'll look at the cell cycle. We'll describe the main events in each phase of the cell cycle. So the cell cycle is a repeated series of many events that a cell undergoes. So one cycle of cell division will have a whole lot of different things happening in it. But cell division is only a very small part of a cell's life. Most of a cell's life consists of a phase called interphase, as you can see, which is represented by about 3/4 of this circle. So the cell is replicating its DNA, but it's also carrying out a whole bunch of other functions such as growth. So again, most of the time that a cell is alive, it is in interphase. The G1 phase of the cell is when the cells grow. Protein synthesis is happening. Remember that proteins are the structural foundation of everything in a living organism. So those proteins are constantly having to be created within the cell. That all happens in the G1 phase. We have a G0 phase or G0 phase. During that phase, the cell is continuing to function, but it's not replicating or dividing. It's actually sort of a resting phase. So most of your cells at any point in time in your body are going to be in this G0 phase. The G2 phase, skip the S phase. The S phase, the cell is replicating. So this is when it's making that copy of its DNA. So it's still not in cell division, but it is copying DNA. And then once it has copied its DNA, it goes into the G2 phase, which is when it's producing proteins that are necessary for mitosis. So this is, imagine it as a preparation phase for cell division. So your cell, again, it tends to be in G0. If there's some growth going on or proteins need to be synthesized, that happens in G1. DNA is replicated in preparation of cell division during the S phase. And then G2 is the final preparation phase where it's undergoing those physiological processes that get it ready for cell division. So when cells leave G2, they enter mitosis, and mitosis is actually the division of the nucleus, and that is followed by cytokinesis, which is the division of the cell itself. So really there are three phases of mitosis. There's interphase, which is sort of that resting phase. Not mitosis itself, sorry, the cell cycle. There are three phases of the cell cycle. There's interphase, and then there is cell division, and then which is mitosis, and then there's cytokinesis, which is the division of the cytoplasm and all the other things that are in the cell. So we're going to look at the steps of mitosis. We have 5 main phases in mitosis, and we're going to be looking at those in detail. Back in the day that I took biology, we only had 4 phases. Prometaphase was not one of them yet, but they decided that there was enough of a distinction between what was happening between prophase and metaphase that we needed an additional step. So in prophase, chromosomes in the spindle form. So remember, the DNA condenses into those chromosomes. So they condense, they become visible in the cell, and the spindle begins to form as centromeres move to the opposite poles. Now, don't get confused with, I said exactly what I don't want you to be confused by. Spindle forms as centrosomes move to the opposite poles. Remember, the centromere is what connects the sister chromatids together, holds them together. And the centrosomes are sort of structural anchoring points for the spindle. The spindle is going to be like finger-like projections that, as we're going to see, they help to actually move the chromosomes in the cell. So prometaphase is the next phase of mitosis. The nuclear envelope breaks down, so now the chromosomes are free in the cell. The spindle fibers attach to kinetochores on those chromosomes. So again, the spindle fibers are finger-like projections, and they attach to those chromosomes. They're going to move the chromosomes across the cell. During metaphase, the chromosomes are lined up along the equator of the cell, so they're still attached to the spindle, but now they are organized and ready for division. In anaphase, the centromeres split. So remember, the centromeres held the sister chromatids together. They split. The sister chromatids now can separate, and they move to opposite poles in the cell. And notice the spindle looks like it's retracting as it pulls those chromosomes to either pole of the cell. In telophase, the nuclear envelope and the nucleolus have formed at each pole. So nuclear envelope starting to reform around the genetic material that has been divided. And the chromosomes decondense, so they turn back into their loose form, and the spindle is going to disappear. And finally, we have cytokinesis. This is when the cytoplasm is going to split. So in animal cells, the cell forms what's called a cleavage furrow, And this really starts to appear during telophase, and it's sort of an indentation in the cell, so cleavage furrow. And then eventually that is going to cleave, which is why it's called a cleavage furrow, and divide into two separate cells. So as it deepens, the ring of proteins beneath the cell membrane that is starting to form, it solidifies and it contracts, and the membrane now becomes two. In plants, oh, let me go back. I thought that was on another slide. So in the plant cells, you can see that there's a cell plate that forms, and two cell walls will actually form, and then cytokinesis is complete in that phase. And those cells in a plant cell, remember that they are organized kind of like a brick-like structure. So the cells have divided into two separate cells, but they still remain adjacent to one another. So here's a question for you. In some fungi and slime molds, mitosis may occur without cytokinesis. What would you expect to find in these species? Well, if mitosis occurs but the cytoplasm itself never divides, we would expect that the cells contain multiple nuclei. If the cytoplasm didn't divide, then even though the nucleus, you have two now, but it never divided, so they're both in the same cell. What is the role of the spindle in mitosis, and what would happen if it could not form? Well, I talked about the spindle being sort of the anchoring point to actually coordinate the movement of the chromosomes to the poles of the cell. So if that did not happen, if the spindle didn't form, then as the cell divided, you might have a few chromosomes here, a few chromosomes there, and there wouldn't be any organization, and the cell would not end up with an exact copy. So big picture in mitotic division or mitosis, the important take home is that you start with a single cell, the genetic information is duplicated, those chromosomes divide equally so that half the genetic information goes to the original cell, stays there, and half of it goes to the new cell that forms. So you have two exact copies or clones of the original cell.